Okay, why I take this question at the end of the lesson? Because I want to give you a message. It is not important that we are going to do this question, because each time I take a question to give you a message, the message is what we have learned, the words, the meaning, definitions are really important to answer the questions. Because I clearly know we cannot do questions unless you study it well. If you study everything well and you can quickly recall everything well, then you can answer the questions. Because when you study, large amount of information go into the brain. And where all the information going into the brain, there is kind of an algorithm the brain used to store this energy to the information. So brain use some method to store this information. Then you try to take this information back again, whether to accept this sentence is correct or wrong. Because you take the sentence and you check with your brain's information whether this is correct or not. This can be only done first if you transfer the information to your brain. If you transfer, then you can take it again. So again, when you take it out again, depend on the practice. So past papers are likely to practice how quickly you can take information from your storage. Chlorophyll absorb violet, blue, and red light. So I clearly told in summary, chlorophyll absorb violet, blue, and red light. And I mentioned previously in the light absorption spectrum, we have seven colors, but 400 nanometer towards the blue and violet, and the 700 towards the red color. So then we have two colors, basic colors chlorophyll can absorb. Chlorophyll B is the main light capturing pigment in plants. I told many times chlorophyll A is the light capturing pigment, main light capturing pigment. And I mentioned chlorophyll A is very important because it can absorb highest amount of light, main light capturing pigment. Second thing, involved in light dependent reaction. Then third one, chlorophyll A is the most efficient for capturing green light. That is obviously wrong because green light is not absorbed. Green light is emitted. It is emitted. That's why we can see green color plants. Chlorophyll A is involved in absorption and dissipation of excessive light energy. Now you see, after 2.5 years of your work, and you study so much of things, it is only one word that you had to replace here. Like, it is not chlorophyll A, it is carotenoid, is involved in absorption, dissipation of excessive light energy. And this word is only A. So, chlorophyll A is the main light capturing pigment in plant. So, chlorophyll A is most efficient for capturing green light. So, it should be replaced as a blue and red light. Photosystem 1 absorb 680 nanometer wavelength. I said this one differently. I said there are two type of photosystem. Photosystem 1 is 700, it absorbs 700 range. Photosystem 2 680, it absorbs 680 range. Now, if you look at this question, only the highlighted words, rounded words are the wrong things. 680 should replace with 700. Chlorophyll A should replace with carotenoid. Here blue and red, and here simple A. So the message is, when you see a question, you see the choices are same because only minor points they changed. It's for them. It's very easy to make question. They copy paste the text from the resource book and then make the sentence, and what is in the below they make one two three four, just change slightly, only one or two word change. So when you see the question, you see everything is like same. How we can handle the problems? First of all, you have to transfer the information to your brain. After that, it is not enough. We have to exercise. So when you more and more and doing question, you quickly get, this is wrong, this is wrong, this is wrong. Like that, information is coming to your brain. Because the incorrect statements have minor change, very small change, and sometimes uh, the one word change, that word is very similar. That word is also very similar to the correct answer. So that's why resource book help you a lot. At least good thing is they just copy paste everything from resource book and change, because this word like a dissipation, the heat is distributed like things without preventing damage, like all are coming from the same, right? Okay. Anyway, we learn clearly today, chlorophyll absorb violet, blue, and red light. That's a correct thing, right? And the next one, we start light dependent reaction. Light dependent reaction, linear electron flow. How we can understand this? When light is there, there is a reaction. In that reaction, electron flow only one direction, because this word opposite is the cyclic, linear and cyclic. So simply we can understand, there is a light. So we learn a lot of things about light. So when the light hit, something is happening. There is a reaction, and the reactions, electrons are moving only one direction. So if the electrons moving one direction, maybe at the end, these electrons taken up by someone, and that is end. So by looking at the word, we can get the idea. Sunlight provide the energy, then the reaction take place, and electrons are moving forward. So we're going to learn this step by step like this. First step. In the first step, I draw here photosystem 2. This is the photosystem 2. So you have learned these are the light harvesting complex, and the middle is the reaction center. So it is consist of pigments, chlorophyll A, chlorophyll B, and carotenoid. In reaction center, we have a primary electron acceptor and chlorophyll A molecule, pair of chlorophyll A molecule. And this photosystem is located in the thylakoid membrane. Okay, let's see what happened now. The light harvesting complex absorb the light. Chlorophyll A absorb light, Chlorophyll B absorb light, carotenoid absorb light. Carotenoid also distribute the light without damaging the systems and the molecules, and it dissipate and it has a photo protection. Finally, light energy taken up by the pigments. So when light energy is taken up by the pigments, all this energy move, all the energy channel flow to the reaction center. Where is the reaction center? Reaction center is in the middle. So what is happening in the reaction center now? This is a chlorophyll A molecule, pair of chlorophyll A molecules. And all the light now channel to this chlorophyll A molecule. The electrons are excited. Electrons are excited. The electrons start to move out of these molecules, called excited. Light energy captured by the pigments, and then the electrons are excited. So after electrons are excited, these electrons move into the primary electron acceptor. So it was here, primary electron acceptor, which is still in the reaction center. So the electrons are moving to the primary electron acceptor. Light energy is absorbed, electrons are excited in the reaction center, chlorophyll A molecules excited with light energy, then the electrons excited, electrons are moving to the primary electron acceptor. You need to remember one important thing. These electrons are rich in energy. Just to indicate the energy, I highlighted yellow color. So these electrons has energy. Where is this energy? It's from the sunlight. Because you know one thing, we cannot destroy energy. We cannot create energy, only we transfer energy. The sunlight energy transfer to the chlorophyll molecules, chlorophyll molecule transfer to the electrons. Now electrons moving with lot of energy to the primary electron acceptor. And this system is photosystem 2 or 680 nanometer range. So light dependent reactions start with not PS1, it is starting with PS2. So now the electrons, they are really rich in energy moving to the primary electron acceptor. Now if I draw it a little bit easy way again, this is a PS1, sorry PS2, thylakoid membrane. Okay. Okay, you can see in the thylakoid membrane there are another three structures. There are another three structures. We don't want to know the names of the structures. We generally can learn them with one word. So we can tell them one word, electron, a carrier protein or complex. So this is electron carrier complex or protein. This is electron carrier complex. This is electron carrier complex. Now in PS1, the electrons accepted the primary electron acceptor, and after that, this electron moving like this way. First electron carrier proteins, the second electron carrier proteins, and third electron carrier proteins. So I try to draw this way, the line, right? Now the electrons are moving from high energy state to low energy state. So this has a lot of energy when it released from this photosystem 2. But when it comes to here, it goes to the low energy state. So what happened to the energy? So we have to find what happened to that energy. So this energy is used to pump hydrogen ion or protons from the stroma to the inside of thylakoid. Energy is used to pump hydrogen ion because this act as a transport molecule in the membrane. Hydrogen ion cannot cross the membrane. Only this protein helps to bring it, but uh, that need energy. So hydrogen ion cannot move easily without energy. Only if energy is used, then it can pump. Then the hydrogen ion or proton pump from the thylakoid into the thylakoid from the stroma. Because if you look at the chloroplast, thylakoid is like this, and inside is here, this side is the stroma. So as it continues, uh, then it pumps large amount of hydrogen ion. So this process continues, light is absorbed, then the electrons are excited. With that energy, it's moving to the electron carrier complex, and then hydrogen ion pumped in. Now, what's going to happen to the photosystem 1? So, photosystem 1 is here. So, before we come to photosystem 1, we have another small issue. Issue is, can you remember when I tell you reactive oxygen molecules? If molecule lost electrons, it is not stable. It is not stable. So we have seen electrons are excited from chlorophyll. Then the chlorophyll molecule are not stable because it lack electrons. So molecule cannot exist like that way. There should be a way of bringing back electrons to this molecule. So in photosystem 2 is very unstable now because the molecules lack electrons. As a result, what is happening in photosystem 2, the enzymes and other proteins in PS2 take water molecules, and this water molecule split into hydrogen ion, electron, and oxygen. Hydrogen ion, electron, and oxygen. So I can balance the equation like this. So this is called water splitting. But Campbell biology book and the resource book use this word as a water splitting, but most of the time there is a standard word for that. Even exam and many textbook use this word, which is called hydrolysis. This is also a very important word to remember. It is called hydrolysis, water splitting. So what is important of that? Now you see one important, in photosynthesis we get oxygen. So where we get oxygen? Oxygen we are getting from water. We get oxygen from water. And what happened to this uh, electron? This electron can move back to the PS2 complex. This electron donate its electron to the chlorophyll. Chlorophyll become again stable. Chlorophyll becomes stable because this electron coming from chlorophyll by the reaction of water splitting. And this hydrogen can still contribute to the developing hydrogen and ion here, protons, or maybe it is again important for another reactions. So we'll see what happened to these hydrogens. After that, we will move into the PS1. So just to summarize it again, light absorbed by PS2, chlorophyll absorb light and electrons are excited. The electrons are moving down its energy level to high energy state to the low energy state through electron carrier proteins in the membrane. That energy is used to pump hydrogen ion in and become a stable the PS complex with the electrons. Water splitting take place, so hydrolysis or photolysis. So it's also photolysis. I think this word is the most important word, right? The standard word, photolysis. So photolysis is the correct word because hydrolysis is again another word we use adding water molecule we can break the bonds. So it is not the correct word. Photolysis is the correct word. But the photolysis word is not used by the book, but in exam it is used. Okay, now we'll see what happened to the PS2 complex, PS1 complex. PS1 complex can also absorb light. And same story is happening, light harvesting complex and the reaction center. And everything is now moving down to the reaction center. The reaction center electrons are excited. So electrons again released from PS1 complex. Now PS1 complex become very unstable because it lost electrons. Same thing happened to PS2. Electron removed from PS2, it's very unstable because it has no electrons in some of the chlorophyll molecules. Now this is a story is a little bit easy for this one. This electron can move into the PS1. There is no proteolysis or water splitting here. This electron coming from PS2 moving to PS1. So then I think we can conclude the first part because everything done. What happened? Oxygen formed and hydrogen ion pumped in. So this is the first thing happened. This is the second thing. So what is the outcome of PS2? PS2 it produces oxygen and it's also pump hydrogen ion in. Then we try to see what is the effect of PS1. What happened to the electron of PS1? This electron again rich in energy. It is in a high state of energy. PS1, thylakoid membrane. Again, you can see three complexes like before. And what are those complexes? Those complexes are electron carrier proteins. And we need to remember name of this one because it is an enzyme. This enzyme is called NADPH dehydrogenase. NADPH dehydrogenase is the enzymes. Okay, now what is going to happen to these electrons? These electrons excited from the PS1 or 700 nanometer move into this complex, move into this complex, and move into this complex. But if you look PS2, photolysis happen, hydrogen ion pumped in. Here, there is no photolysis, there is no hydrogen ion pumped in. That means this electron reaching to this complex with energy. So this has still has energy. Energy did not lost. So no loss of energy. So where does energy going now? Where does this energy move? So where can this energy move? Okay, we have another reaction, important reaction here. NADPH dehydrogenase, it's an enzyme. This enzyme catalyzes a reaction. The reaction is this electron, proton or hydrogen ion, plus a molecule called NADP plus. Nicotinamide, adenine, dinucleotide, phosphate. This act as a cofactor. We learn enzymes and cofactors. Then this molecule NADP act as a cofactor and it together with these enzymes involved in the reaction, and after that this NADP plus become NADPH. So I write here maybe easy NADPH, because this I removed NADPH. NADP plus is in oxidized form and it becomes its reduced form. Okay, what happened to the energy? Now energy is captured by this molecule. Because these electrons has the energy, it doesn't lose. This energy is moving to NADPH. NADPH is an energy rich molecule. NADPH, I write here, NADPH is energy rich molecule. So this is a product. We can consider NADPH is the product of light dependent reaction. It produces oxygen. It produces now NADPH. Yes, I think we have almost completed the reaction. This electron move into the PS1, and PS1 electron now move to the NADPH. NADPH is energy rich molecule, and this oxygen can diffuse out of the leaf or the cell. Only the unsolved problem is what happened to this hydrogen ion pumped in, because uh, the energy of this electron create this hydrogen ion gradient. Like stroma has less proton, inside has more protons. The energy is now in this place. Electron energy transfer to increase hydrogen ion concentration inside. So energy is with the hydrogen ions now. And in PS1, it is less complicated. Electron moving to NADPH dehydrogenase complex, and the energy rich electron incorporated into the NADPH molecules. NADPH has energy. NADPH is energy rich molecule. Okay, we will now look what happened to this energy which is now within the protons or hydrogen ions. Okay. This is the thylakoid membrane, part of the thylakoid membrane, And so far we have seen the story starting with PS2 complex and here PS1 complex. So when the light absorbs, the electrons are excited, electron is moving down and coming to PS1 complex. So hydrogen ion pumped in to the thylakoid. The energy is used from these electrons. And PS1, again the light is absorbed, then the electrons are moving and finally it produces NADPH. And in this reaction, electrons is again produced by water splitting and this electron is going back to this system. So what are the product? Oxygen, NADPH. Okay, next. In the thylakoid membrane, there is another enzyme complex. So this enzyme complex is located like the in thylakoid membrane like this. This is an enzyme. The name of the enzyme is ATP synthase. ATP synthase. Okay, so what is the picture now? There's a large amount of protons accumulate inside the thylakoid. Outside less, inside high. So we use a name to indicate in this kind of a situation electrochemical gradients. It is just like electricity when we have electrons one end, other electrons, less electrons. Then if we activate electrons to flow, electron is flowing from high electrons to low electrons. So something like this, we have a high amount of hydrogen inside, low amount of hydrogen outside. So this hydrogen make a potential, hydrogen make a energy level, this hydrogen ion make a gradients called electrochemical gradients. That means the energy is trapped in this place. Because electrons energy, this electron energy is used to pump. Then that energy is transferred to this to create this gradient. So we call it electrochemical gradient created by the proton or hydrogen ion. So hydrogen are separated by a membrane, one side less, other side is high, then it creates a potential gradient. So this potential gradient is really powerful and it has so much of energy trapped. But we can't keep hydrogen ion like this in the membrane because you can understand the pH can also change.